just generally increase bonding and increase like all of these sort of good feelings. It has a specificity to it so that oxytocin increases the subjective attractiveness of your partner, right? And so we can see this is males and the other figure is females. And in both of these cases, the adding of oxytocin makes your partner more attractive, right? This is males, this is females, and again, we see the same situation. So oxytocin increases your subjective rating of your partner's attractiveness. It is not a universal thing, and so we can see this in both males and females, that if you show someone who's in a pair bond, their partner, they rate them as more attractive than they are generally rated by the public, but also, and I think this is the other kind of key feature, they rate everybody else as less attractive, right? So you're getting it on both ends, which I think it doesn't take a lot of sort of thought experiments to realize how this solidifies pair bonding, right? And social bonding in general. Then I think this is where we ended last time, if I'm not mistaken. So for all those dog people out there, this is a demonstration that, number one, from the human perspective, having a dog and that interaction with the dog, right? When you come home after a hard day, maybe it's been raining and a couple of tests, not today hopefully, but but you had a really hard day, bad things happen to you, you come home and the dog is just overjoyed to see you and it makes you feel better, right? I feel better now, that's oxytocin release, right? So I don't think that's surprising from here, but I think one of the things as a dog person myself that I find encouraging is that long gaze dogs, the dogs that will actually look at you in the eye, which is again most dogs that we keep around us in large numbers, they also get an increase in oxytocin from interacting with us, right? So it really is a two-way relationship. They really are excited to see you, and they feel they are getting a release of oxytocin as well. This has never been done in cats, and I think so I think it would really vary depending on the cat, right? So I don't know how you would classify them as long gaze, short gaze. I think most cats are like no gaze, right? Kind of like the wolves. But I think you get the idea. Now, this can kind of be summarized, right? This is, showing this kind of same basic idea. This is just with dogs. I don't think I showed you this figure last time. And so, essentially, they are giving the nasal spray to the dog and allowing them to interact with their human companion or to interact with other dogs, okay? And so one of the things I don't know if I've mentioned this before or not, but oxytocin, when it's given to a rat or a vole, as we'll see, this is typically an ICV, right? So intracerebral ventricle, like it's given directly into the brain. And the reason that is so is because it's a peptide. If you take it orally or you put it in the bloodstream, it's going to get chopped up very quickly. So to be able to get the drug where we want the drug to be, it's a direct injection. Now, you're not doing that to pets. As a general rule, if you can imagine, like, oh, are you interested in studying oxytocin in your pet? Okay, great, yeah. So we're going to drill a hole in his or her head. We're going to stick a cannula down there. No. So same thing with humans, and this oxytocin therapy is used in, there's a lot of work being done now in autistic children. So basically, if you know much about autism, one of the hallmarks of autism is challenges with social attachment, right? Challenges with parental attachment, challenges with any kind of social or romantic bond. So autistic children in particular have a very difficult time bonding with their parents. And so over at Emory, there's an institute on autism that uses oxytocin to treat autistic kids with oxytocin, right? And so the idea is that you have to get it into the blood and preferably even into the brain. And so you have to, or they've developed an intranasal spray, so something you spray into the nose of the child, adult, or dog in this case, right? So this is a intranasal injection, and then what we're gonna do is just measure the rate of affiliative behavior during the observational period, right? And what we see is that I should say, what do we see?
More affiliation with other dogs.
Yeah. The dogs are more affiliative with their human companions and with other dogs in the presence of oxytocin, right? So another way to sort of look at this, oxytocin makes individuals more affiliative, right? I think this is a sort of general pattern that we're going to see. Okay, so we're going to transition now to our experimental models, okay? So our experimental models for social behavior are, by and large, just the voles, right? So we're not going to use rats, we're not going to use mice, by and large. We're really going to focus just on voles. This is a sort of standard model for social behavior. And to sort of set up some of the generalities of the vole model, prairie voles are monogamous. And if the vole starts with the letter M, it's not monogamous. So the way to think of it, these are promiscuous voles. And that monogamous versus promiscuous, I think, doesn't really doesn't really explain what's going on, right? So another way to think about this, perhaps a better way, is that prairie voles pair bond, right? Prairie voles pair bond, and meadow voles don't, right? Meadow voles and montane voles are not that social. And so to kind of give you a sort of general idea of monogamy in animals, this is sort of the classic. And if you remember the prairie voles from the Coolidge effect, right, if you you've got one of these, we'll just say this is the male, and you've got the partner female, and then you've got a stranger female. So this is a 3-chamber test, okay? So the way that this works is that the females are essentially tethered, right? Means they're like with little leashes that are attaching them so that they can't go into all 3 chambers. We have a previously mated or previously bonded female and a stranger female, okay? Now, under typical conditions, if you did this to a rat, right, where's the rat gonna go?
Stranger.
To the stranger female, right? The rat is almost always gonna choose this one, right? And this is sort of, prairie voles are always, as we'll see some data on this, pretty much 90% of their time is gonna be spent here with their partner. And again, if you remember the Coolidge effect, right? We looked at this is if you show a potent novel sexual stimuli, you can reduce the relative refractory period. So in prairie voles, that still works, but the odd part is the novel female makes the prairie vole want to remate with his male, or female, soon, right? Okay, so now, if we look at this, essentially, if we graph this for the prairie vole, right? Well, these are prairie voles. If we graph this, this is partner, this is stranger, and this is, we'll just say, cuddling time, because what they're actually measuring, they call it huddling, but I really feel it's a missed opportunity to call it cuddling. But it's literally physical contact with your partner, right? If that's not cuddling, I don't know what else is, right? So, they spend all this time with their partner and very little with the stranger. So this is the prairie vole. Any of the M voles, right? So this is partner, this is stranger. It looks like this. Now, what's going on here? Well, the idea is that prairie voles, as I said, prairie voles like to socially bond. So they will go over and bond with their mate, right? As opposed to this stranger. Meadow voles don't like being in the same compartment with females for very long. So they spend almost all of their time in the center compartment, right? So it's not as if they're just going, right? It's not as if the prairie vole spends all its time with its mated female and the meadow vole spends all its time with a stranger. It's actually that meadow voles don't like to spend time huddling or cuddling with either females or any females at all, right? So let's take a brief look at their brains. As I said, we talked about this being a story about nonapeptides, right? Now, this is an autoradiograph of oxytocin receptors, okay? So remember, again, sort of the way this works is you are putting in hot oxytocin onto a section of brain, which is just labeled with something radioactive, or by hot, we mean we're just attaching like a hot phosphate or something to it. We are putting it on here and then we're putting film over it, and the darker the area, the more receptors are there, the more oxytocin receptors are there. And so what we're going to compare here is the prairie versus the montane vole, okay? Now, do both of them have oxytocin receptors? Yeah, okay, so that's the first thing to notice, right? They both have oxytocin receptors, and pretty reasonable amounts of oxytocin receptors, but the key element here is that they are in different places, okay? Now, what determines where your receptors are? Genes, okay? So what part of the gene? Is there an answer? Just stop answering there. Okay. didn't want to. Okay. So, all right. Remember how like we talk about like alleles, right? Remember we get one allele for a gene from mom and one allele from dad. But notice that most of the time, and so the definition of what is a gene?
It's code.
Code for
Protein.
For protein, right? Yes. Okay. So some fundamentals, right? So, would you guess that the oxytocin receptor between the prairie and montane vole is identical?
Sure.
Yes. Right. So the coding region of the gene is identical, right? It makes the exact same receptor. So again, it's not that the receptors are different between prairie and montane voles, it's that their distribution is different. And so when we say, how do we determine where these things are expressed? Yes?
Is it like the promoters that are on the
Yes, it's so promoters or the cis regulatory regions, or maybe a better way to sort of say, right? So if you think about an allele, right? So if you've got, if this is, let's see, OTRA, right? So oxytocin receptor A. It's these regions here, right? These non-coding sequences that precede generally the gene itself that determine where that gene is expressed and how much of it is expressed, right? So, and they've been at this story for a long time, and it turns out that there are certain microsatellite repeats that are different between prairie voles and montane voles that can be identified to explain why, from a genetic level, you have this different receptor expression. And so that's just kind of a kind of important thing to keep in mind, right? That we are talking about the exact same receptors located in different places, because the genes that control, right, the promoter, the cis regulatory regions that control where those genes are turned on are obviously going to be different between these two species. Okay, so looking at this chart, do any of these regions stand out to you as being associated with something? The nucleus accumbens is associated with what? Right, so this is reward and reinforcement, pleasure, right? So that's one big area. Which ones have oxytocin attached to the nucleus accumbens in large amounts?
Prairie.
Prairie voles, okay? Now, are any other regions that show? The amygdala, what's the amygdala? Fear, okay. Remember, what did we say oxytocin did? It's an antidepressant and it's an anxiolytic, right, as well. Antidepressant and anxiolytic. Where, if you're anxious, what region of the brain do you think is going to light up a lot? The amygdala, right? So notice that in our prairie voles, we have two regions that we can sort of almost automatically associate with behavioral changes, right? Now, notice that places like the lateral septum, where we have lots of expression in the montane vole, right, much more than in the prairie vole, says that it's certainly not that these animals don't have these regions, right, that they don't have these receptors, but notice how shifting where the receptors are could conceivably give us very different behaviors, right? Okay. So let's kind of explore this a little bit more. So this is a complicated figure, but it is one that is essentially really critical to our general understanding of this model, right? So the idea is the top is females and the bottom is males, okay? Now, I just wanna, female, I, so oxytocin is females, generally pair bond. The molecule is oxytocin, and for male pair bonding tends to be more vasopressin dominated. It's not to say that females don't have vasopressin, or that males don't have oxytocin, or that they don't play a role in social behavior, but in this model you see a divergence between the sexes into the sort of major neuropeptide that affects these processes. So oxytocin for females, vasopressins, yeah, vasotocin, because it's a fish. In fish, it's vasotocin. Okay, this is in males, right? So notice that it's not that they don't have oxytocin receptors in the nucleus accumbens, it's just that they principally use vasopressin for this behavior. Okay. Now, what we are going to be doing here is giving an antagonist, right? So OTA, see on the top there, oxytocin antagonist, right? Down here, if you can't see it, what this says is V1ARA. So this is a vasopressin 1A receptor antagonist, and the top panel is being given an oxytocin antagonist. All right. If you remember, I said that these are things that we want to put into specific regions of the brain. What we don't want to do is, so if we give something to, let's say, the third ventricle, it means it's going to go everywhere in the brain. But what we're interested in, if we can sort of think about this, and I'll sort of jump back to this slide for a second, is that notice we have all of these places that have oxytocin receptors, but what we're interested in is where the oxytocin receptors that are responsible for the behavior we're interested in, pair bonding, right? Where those receptors are. Okay? So we've got a whole brain to choose from. These three areas on the top, we're just going to do females first because we've been talking about oxytocin. Okay, so we are going to put a cannula, which is basically, we're gonna drill a hole in the skull, we're gonna drop a needle, and we're gonna put that needle's output into the nucleus accumbens, the prefrontal cortex, or the caudate putamen, places that have high amounts of oxytocin receptors, okay? Now, we're going to give each vole 2 treatments. Those 2 treatments are drug or vehicle. Okay, what is vehicle in this case? It's not quite saline. Close. CSF. Right? So if we're putting something in the brain, right? CSF is cerebrospinal fluid, right? So it is kind of ish saline, but with some little extra stuff in there. But so the idea is that we're going to give, right? Yeah, we're going to give the drug, or we're going to give saline into, or see, you've got me doing it now, CSF. We're going to give CSF or the drug. Now, what we're measuring is the time spent in physical contact, this cuddling behavior that we see, okay? So are we measuring oxytocin? No, okay? This is a behavioral output, so we're either giving the antagonist or vehicle, and we are putting them in this environment, and we are then asking how much time they spend with their partner versus the stranger. Now we can just switch the sexes around and it's exactly the same for males and females. Now, for the males, we're doing the same thing, but instead of using an oxytocin antagonist, we're using the V1AR antagonist, okay? So, what is combined CSF, would you suspect?
Regular CSF? I don't know.
With their partner?
No, okay. See, I told you this figure was complicated. Now, notice that each of these animals, or groups of animals in this case, right? Everything is never a single animal, it's always a group of animals. But notice that there's a group of animals that have a cannula in the nucleus accumbens. There's a group of animals that have a cannula in the PFC. And there's a group of animals that have a cannula in the caudate putamen. Each of those groups gets both treatments, right? So, what combined CSF is the control given to the nucleus accumbens plus the control given to the PFC plus the control given to the caudate putamen. So, all of the control treatments for all of the brain regions that have cannulas combined together. So it is, right, a summation of all of the control groups. Now, notice that our prairie vole females spend about 70 plus minutes in physical contact with their mate when given CSF. So they're over here almost half of the 3-hour period where they spend, what, about 15 minutes or so over here.
Mm-hmm.
Right? Does that make sense?
Mm-hmm.
Okay. So that's the normal case for female prairie voles. And you can see the exact same thing in males. Males do the same thing, right? Which is what you would expect, that these numbers are going to be pretty much right on top of each other because, you know, that's what they do. Okay? Is this clear to everybody? We'll go over it again. I know it's a little bit challenging. So now, we are breaking out the individual groups. So all of everything past this dashed line got the antagonist to the specific brain region only.
Mm-hmm.
So, what we're looking at is the effect of the antagonist given in a specific brain region on the behavioral output of the animal. So if we give the oxytocin antagonist to the nucleus accumbens, what happens to the behavior?
It swaps.
It swaps almost, right? It switches from a preference, a strong preference for the partner, to really a pretty strong preference for the stranger. All right? So what does that tell us about oxytocin receptors in the nucleus accumbens?
There's a lot in there.
Well, there's certainly a lot there. Hmm? Well, they're really critical. Without them, do we get pair bonding? No. Now, again, remember, is the animal still going to release its normal amount of oxytocin in this interaction? Yes, we're just blocking the effect of the receptor, and we're only blocking it in one place. So all of the other receptors can still respond to oxytocin. So notice that it's not just that they're involved or that there's a lot of them, it's that they're critical. So without those receptors, they will not exhibit the behavior normally. Right? Now, we're going to, just for so that we can sort of be reading these correctly, we're going to go all the way to the other end, to the caudate putamen here. All right? Now, what happens when we give the oxytocin antagonist to the caudate putamen?
Increased.
Yeah. Maybe more time, but at the very least, nothing related, nothing that inhibits pair bonding.
Right.
So we know there are oxytocin receptors there, but they don't contribute to pair bonding. Right? Because if you block them, nothing changes. Yeah?
So wait, they get the oxytocin antagonist in all three areas, right?
No. Okay. So there's one group of animals that gets the oxytocin antagonist to the nucleus accumbens.
That's what I
There's
mean. The three different groups, they all get the oxytocin antagonist, though,
Yes.
right? Okay.
Yes, they all get the antagonist,
Yeah.
but they only get it in one part of
In
the
one
brain.
place, right? I
Yes.
got you.
Yeah. Okay.
Yeah.
So this tells us, and is the prefrontal cortex affected by oxytocin?
Yes.
I'd say yes, right? It definitely changes the pattern. I would say it's not quite as dramatic as the nucleus accumbens, but it certainly does change it, right? So we can say, or what is the takeaway here?
The nucleus accumbens is critical for pair bonding.
I think yes, we're going to build on top of that, right? So the nucleus, what in the nucleus accumbens is important for pair bonding? Oxytocin receptors in the nucleus accumbens are absolutely critical, right?
Females.
In females for pair bonding, right? And I would say to add the receptors in the prefrontal cortex play a role, but not as significant a one. Right? Because there's definitely an effect there. It's just not as dramatic. And now, notice, the other thing that we can sort of think about is when the receptors in the prefrontal cortex are blocked, the receptors in the nucleus accumbens are still working. Right? Just like when the receptors in the nucleus accumbens are blocked, the prefrontal cortex and the caudate putamen and everywhere else is still working. So notice how much stronger the effect, right? If you eliminate the nucleus accumbens ones, you eliminate partner preference altogether. Right now, I want us to stop and think about that for just a second. Like, I know we're like, okay, yeah, that makes sense in like a pair bonding perspective. So, if you gave an oxytocin antagonist if you took an oxytocin antagonist in your nucleus accumbens and went home to your partner, what would you feel?
No.
Nothing, or potentially, I don't want anything to do with this.
Yeah.
Right now, notice that what reinforces social behavior? What reinforces your interactions with the people you care about? Oxytocin, and the interaction of them that generates positive feelings, which is oxytocin and dopamine. If you block those things, right? Again, we think of our attraction to people, we think of our bonds, we think of our friends and our families as things that we've almost sort of cognitively decided, oh, we love that person or whatever, right? But notice that if we give a drug in the right place, that we are going to change the feelings that we have. And we might realize, oh, that's still our mother, or that's still our, boyfriend or girlfriend or whatever. But notice that the feelings are driven by oxytocin, and that they can be blocked if we find the specific regions are necessary. Okay? Does that kind of make sense, everybody? Now, the same general idea here is found in males, right? So we've got the ventral pallidum, that's the VP, the medial thalamus, and the medial amygdala are the two other regions that we have lots and lots of vasopressin receptors. Where's the important place for pair bonding in males?
Ventral pallidum.
The ventral pallidum, right? And so if we block the receptors there, right? Again, remember, we're not changing the amount of vasopressin, we're just blocking the one brain region from responding to that. And again, we see it's the ventral pallidum. Does this make sense, everybody? Okay. Right, it's kind of a, yes?
Is the combined CSF like your control?
Yes.
Okay.
Combined CSF is the control, right? Because now notice that, or I should say, what are you controlling for? Hmm? Yeah, you drilled a hole in their head, right? Dropped a needle in there and are supplying the brain with fluid, right? And we're asking, right? Because again, if you just had an awake behaving animal that had nothing, right? Someone's gonna look at that and be like, well, yeah, okay, sure, fine. But they've got a cannula in their head and they don't.
Mm-hmm.
But so also notice that each individual is serving as its own control in a way. So the same animal that, right, now let's imagine the voles, these are the females in the nucleus accumbens group, right? They're given saline and they just spend all of their time with their partner. Then a week later, they're given the antagonist and lo and behold, same partner, stranger, they go and spend all their time with the stranger.
Mm-hmm.
Right? That's essentially what happened. And so on trial 1, with the control, is going to be included in the combined CSF. Trial 2 goes just in the nucleus accumbens category. Yes?
So could you explain why, if we put oxytocin in the nucleus accumbens for females, like, in my head I would think that they would be with their partner more because they're feeling.
Isn't it oxytocin?
It's the oxytocin antagonist.
Oh,
Yes.
never mind.
Well, so
That makes
yes,
complete sense.
100%. If we gave them oxytocin, our idea would be they should just be like even more over there. If we gave them oxytocin instead of the antagonist, you might see it's up to, it might even be all 3 hours. They're just like over there, like, oh, it's been so long since I saw you in the cage this morning, right? Kind of deal, right? But yes, exactly. This is the antagonist. Yes.
How do you explain the increase when you give the agonist to the caudate putamen?
Well, so the first thing to note is that we're talking about a difference of maybe 10 minutes over 3 hours. So
Okay.
the first thing that we
Mark.
should look at is, right, and again, notice that for a lot of these figures, and if you read the book, you can kind of see this, is that they're not putting, unless the asterisks are relevant to the question, right? If you went and looked at this figure, if you went and pulled the original paper, you'd see these analyses. My suspicion is that that's not a significant difference.
Okay.
And so even if it looks different and it's not statistically significant,
Yeah.
right? If you remember from our the first assignment we did where we had A and B and C columns, and if they were the same letter, even if the bars were of different height, we would say they're not different,
Yeah.
right? And so that would be my suspicion here, right? And if not, we could then design a set of experiments, say, okay, well, maybe they're contributing in some other way, but that would require us to have a separate set of experiments to learn that data, right? Yes?
So it would be pair bonding in females is mediated by oxytocin receptors and nucleus accumbens.
Yes.
And pair bonding in males is mediated by vasopressin receptors in the ventral column.
Yes.
Okay.
Yes. Got it? Yeah, a great summation, actually. A nice little bow on our discussion. All right. So, if we look at this, notice that our oxytocin receptors, let's see, which ones are the prairie voles?
Both of them. Oh wait, prairie voles?
Which are the prairie voles? Well, I guess we should say left or right.
Oh, right.
Left. Left.
I don't know. I can't. That's
Okay.
What does the dark mean? It's a
All right, so
concentration.
let's go through what this is, right? This is right when you guys are sitting in lab meetings and you get figures and all this. This is the important things of being able to look at this and based on what we know. So this is an auto
Left is
radiograph, right? This is right. So the darker means what?
More.
Higher receptors. And so the top panel is oxytocin receptors. The bottom panel is vasopressin receptors. So,
Left
where
is prairie vole.
are the oxytocin receptors in prairie voles?
Nucleus accumbens.
So which one is it left or right?
Left.
Left is the prairie vole,
For sure.
right? Where is the important region for males with vasopressin receptors?
Ventral pallidum.
Okay, so same thing, prairie voles on the left, and we don't even have to have this labeled, right? So notice that we could look at this and be like, okay, well I see a lot of dark oxytocin receptors in the nucleus accumbens, that's gotta be a prairie vole, that's gotta be some other kind of vole, right? Notice the receptors are there in the PFC. Notice here also notice those three regions we looked at. PFC, Claudia putamen, nucleus accumbens, all right there. High expression of the receptor in all three places. And which are the important ones? Just the nucleus accumbens for this behavior. Now, do those other receptors likely contribute to other behaviors? Yeah, for sure, but not this behavior. Okay, so let's ask another question here. Does living with someone change your brain?
Yeah.
Oh, all right. I don't know if you're just getting the feel for this or like, yeah, it totally does, yeah. All right, so let's talk about cohabitation-induced changes in the brain. Okay, so notice how inexperienced versus experienced animals, right? We saw this a lot with females in parenting, right? That an inexperienced individual is different than an experienced individual. So someone who is bonded is different than someone who is single, right? And in this case, we're looking at bachelors versus bonded males. But the principle, right, so what happens to oxytocin levels when you enter a pair bond? Increased by about what for humans, for us?
Tenfold.
It about doubles.
Okay.
It's about doubles, not 10 times. It's about 10 times. If it was 10 times, you'd really feel for Valentine's Day would be like a completely different demand, right? Anyway. Okay, so it's about double, and that's kind of an important thing to note, right? So it's about double, and this is just for new relationships, right? There's a sort of a waning that happens, but notice that if we're increasing the amount of oxytocin, we're likely to also be increasing the amount of receptors as well. And so we can actually measure this. So notice that we have, so D1 and D2 refer to what? Does it say on there? What kind of receptors are those? D1 and D2, if you've heard this one before? I got some neuroscience people here. What's D1 and D2? Okay, no. Okay. What do we associate the nucleus accumbens with? Any guess what D1 and D2 are? They're dopamine receptors. There's two main types of dopamine receptors, right? D1 and D2, right? There are also D3s, but D1s and D2s are sort of the classic dopamine receptors. They kind of do opposite things. But D1 and D2, they're both heavily expressed in the nucleus accumbens. But notice that there is a significant difference in the number of dopamine receptors, right, in the nucleus accumbens in bonded males versus non-bonded males. And so we can actually quantify this a little bit better. And so essentially the idea here is that we're going to be looking at dopamine receptors in two different brain regions. And just so we have the nomenclature correct, NACC is what? And CP is?
Caudate putamen.
Yeah,
What is it?
caudate putamen. And so notice that these are places that have oxytocin receptors. Okay? So these are places that have oxytocin receptors and they have dopamine receptors. The only manipulation that we're doing here is whether or not you are a single vole versus a pair bonded vole for 2 weeks, right? So you're in a 2-week relationship with another vole. What does being in a relationship do to your D1 dopamine receptors? It massively increased it. D2? No difference, right? And that's just in the nucleus accumbens, right? So notice that.
Do two voles always bond if they're by themselves? Like, is there like, are they like picky or like, oh, I don't like this one, so we never gonna bond. We just gonna be here together.
So typically, there's two ways I can answer this question, right? It's how they do it in the wild, right? Where notice that all monogamous animals, or I should say all, the basic model for monogamous animals is that they have to pick each other,
Okay.
right? So males are doing a little bit of selecting, females are doing a little bit of selecting. And so in the wild, they sort of, you get to meet a lot of voles, right? And then you just pair bond. In the lab, they essentially just kind of put them together, and if there's nothing else to do, they generally, will bond. Not all of them, but they're usually not given a choice in the laboratory environment. But again, they will bond, right? And I think that there is some logic to this, right? If you left probably 2 people alone with nothing to do for a long enough period of time, I mean, unless it really went sideways, right? You'd have a higher frequency of bonding than you would if you just sort of randomly select.
That's good.
So I thought the nucleus accumbens was for females, or is that just oxytocin?
Well, so in prairie voles, now this is the interesting part. So in prairie voles, both sexes use both hormones, but again, as I kind of said, the primary pair bonding hormone is oxytocin, and as we just described, it is in the nucleus accumbens. Males also, right? And again, I don't want to. It's a great question, right? I don't want to gloss over it, but notice that in these males, so these are both males, right? And you can see the nucleus accumbens up there, right? It's also hot with oxytocin as well in the nucleus accumbens. Now, we know that if you block this in females, you completely abolish pair bonding. That is not the case as in males. If you do this in males, you will reduce, but you will not flip it like this. Right? And they've done that work as well. And so it's not as if, so essentially if you give a male an oxytocin antagonist, you will reduce the amount of affiliative behavior that he will produce towards his partner, but you will not get the flip like we see in both of those things. And that's why there's this, that's why I said it's kind of a general trend to say that oxytocin is sort of dominated by, oxytocin dominates pair bonding in females, and vasopressin does it in males because you can reverse it by just giving the antagonist in one particular brain region. But you do see increases in dopamine receptors in the nucleus accumbens in males as well as in females. So again, it's slightly more complicated, but it's a good sort of general idea here. Okay, so we can actually go sort of one step further, and this is a pretty exhaustive review on the different neurotransmitters, non-peptide interactions that you get in basically unbonded versus bonded compared to prairie versus meadow, right? So this is like prairie versus meadow. This is naive prairie versus bonded prairie, right? And so notice that, again, there's a fair bit of complication here. So if we see socially monogamous, right? Socially monogamous species like prairie voles, and we look at D1 receptors, the ones we just saw, those are the ones that are important for pair bonding. Notice that our socially promiscuous species has more D1 receptors there, okay? But we see a lot more oxytocin receptors in the nucleus accumbens than we do in the socially promiscuous species. Right? I want to pause. I know this is complicated, but so the idea is, do things feel good? Another way to kind of maybe rationalize this, again, it's kind of tough with social behavior because we're such social animals, but do certain things, do behaviors make montane voles feel good?
Sure.
Yeah, absolutely. They have a reward and reinforcement system. It's just that they are not pair bonding animals, so they don't get the responsiveness from social contact like the prairie voles do. So what we see is a greater number of oxytocin receptors in the nucleus accumbens of socially monogamous species, right? We saw that figure too, right? Remember? I know this is a lot of brain slides, but notice the nucleus accumbens does not have any, right, or very low expression of oxytocin receptors. Oh, I'm going the wrong way.
That's right, that's right.
All right. But we can see prefrontal cortex, the bed nucleus, the stria terminalis, and the nucleus accumbens, a lot more oxytocin receptors, right? And many more vasopressin receptors in the ventral pallidum, where we saw this is the primary bonding zone for males. If we look at the naive versus pair bonded, notice that for all of them, we are increasing vasopressin, oxytocin, and D1 receptors in all of the relevant brain regions after being bonded. So the pair bonded brain is different in terms both of its release of nonapeptides as well as the number of receptors. So when you are in a pair bonded relationship and then that pair bonded relationship ends, how do you feel?
Sad.
Why?
Because your body changing. You're not getting the...
Because you've taken away a hormone that causes the release of dopamine. So not only have you taken away the antidepressant, you've taken away the anxiolytic, and you've taken away dopamine, which is your reward hormone as well. So, and are you going to notice this? Yeah, because you have a whole bunch of receptors that are there, that are not being occupied anymore. And so our subjective feeling is, well, that feels terrible, right? So when the bond, whether it's a loss of a friendship, right, where someone close to you dies, a relationship breaks up, notice that the feelings that you have are because this system is no longer releasing all of the hormones that we associate with the positive feelings of social attachment. Yeah? And so when we kind of started, I don't know if we had a test in between there and here, but if we remember, what we kind of started with was this idea that measuring the strength of the pair bond can, or one way to approximate the strength of the pair bond is to measure how bad the break of that pair bond feels, right? Now, again, the idea here being that the stronger the pair bond, the more of all of this that we're getting, and when you take it away, the release, right, the sort of aftermath will be that much worse, right? This makes sense, everybody? So this encourages the organism to do what? Stay together, right? It's like, like that, people start using drugs to feel good, right? But the addict takes drugs to not feel bad.
Right.
Right? And it's the same, notice it's the same systems. Anyway, not to be cynical, but I think it's important for us to sort of see the underlying neurochemistry behind what we sort of think of just these are our feelings, but these are actually things that are mediated by neurotransmitters, and it also explains why things feel good and why the absence of those things typically feels bad, right? Okay. So, this is vasotocin. These are vasotocin neurons, and this is the violet-eared waxbill. I imagine you will agree with me that it's quite the attractive bird. Right?
Very cool bird.
Very nice looking bird. So, just as a reminder, what's CFOS again? It's a measure of activity, right? So anywhere where we see activity, we see CFOS. Now, VTIR FOS plus means that we are double labeling these cells, right? So to be counted as a VTIR neuron FOS-IR plus means that you have to both be positive for vasotocin, right? Meaning that you are a cell which produces vasotocin, and CFOS, which means you are a cell that produces vasotocin that has just been active. Okay? Does that make sense? So this is the proportion of vasotocin neurons that are active after exposure to one of three conditions. Okay? Seeing a lot of blanks.
You said they have different vasotocin after they
Yeah. Yes. So notice that this scale only goes up to 90%, but so basically, all right. Sometimes I think bar graphs hide a lot of the magic about what's going on, right? Okay, so to do this experiment, we're going to look at a specific region of the bird brain that has vasotocin neurons, okay? Because we really we're interested in vasotocin, so we don't really care about brain regions that don't have vasotocin neurons. So what we're gonna do is put an antibody on there that stains for vasotocin, right? So all of the cells that have vasotocin are gonna show up as, let's say, green. I don't have green, so you just have to trust me on black. And so notice that in all conditions, right, we're labeling all of the neurons that have vasotocin, right? That's what sets the 100%. Okay? Now, we're on those same slides. What we are doing is also labeling CFOS. So we have two separate antibodies. If we remember this from the Gallinin neurons, where we have two different colors, and when they show up together, we know that it's a Gallinin neuron that has channel rhodopsin. It's the same idea. So some subset of these versus some subset of these are going to be positive. So notice that, let's say if this is 10, so this would be 40% are positive for 40% of the vasotocin neurons are positive for FOS. Right? Versus this one, how many did I draw here? 6, 7, 8. This is, I feel like I put too many circles in here, but you get the idea. Right? So this is 80% of the total number of vasotocin neurons are active in this condition, and in this condition, 40% of the total number of vasotocin neurons are active. Does that kind of make sense? It's not obvious to glean that necessarily if you haven't seen a lot of these figures, right? So this is the percentage of the total number of vasotocin neurons that are active when you're either in your cage by yourself, in a state with a group of, right, the males, let's say, or your pair bonded partner. So what's going on here? Okay, so first off, what are we interested in?
You want to see more activity?
So more active. So yeah. So we got vasotocin neurons. So what we're interested in is the response of vasotocin neurons to different social circumstances. Right. Remember, vasotocin is a peptide, right? That's involved in social behavior. So we're going to change the social situation and see how the vasotocin neurons respond. Does that make sense there? Right. So we're interested in how vasotocin neurons respond to different social situations. So basically we're going to take our wax bill here, our violet-eared wax bill, and we're going to put them into one of three conditions. Do they like being with the members of the same sex? No. Now we know that now. We can sort of back calculate. They don't like that. That's an aversive social stimuli. Do they like being with their partner?
Yes.
Yeah, I think we can say that they like being with their partner. And notice that this is a bi-directional effect, right? Just being alone in your cage, I was like, all right, it's okay. If you put them in an aversive social stimuli, what happens to the vasotocin neurons?
No activity.
Right, they become less active. And what happens if you put them in a positive social environment? They go increase, right? So the vasotocin neurons respond bidirectionally to positive and negative stimuli. If the same-sex cohort was at like 27, what would the takeaway be?
The same sex.
If the graph,
They don't care.
if it looked like this instead of this, what would the takeaway be here?
No aversion.
Well, it might be that they don't mind being with that so much, but it also could be, notice that it could be that vasotocin neurons just respond to positive stimuli. right? So here, notice that you're getting a response to both situations. So you're getting a negative response to an unfriendly social situation and a positive response to, and again, we're not comparing these two together. We're comparing this to this and this to this, right? Those are the critical comparisons, not so much this to this. If we didn't have this control, would we be able to distinguish whether or not the response was bidirectional?
No.
No, right? Exactly. We need, the critical comparisons are this to this and this to this, right? Because that shows us that our vasotocin neurons are responding bidirectionally. Yeah? Okay.
Oh yeah.
Oh yeah, great. All right, one more, well actually we have two more bird slides. I'm not gonna spend a lot of time with this one. So essentially, notice again that one of our main themes of oxytocin is that it makes you prefer your group, right? Your partner, your friend group, your social group, right? It preferentially prioritizes group over non-group, right? And this is kind of another way to sort of think about this. So we've got we'll start with C. Notice that they have handily colored our birds for us. Zebra finches, I can tell you, they don't really look that different. They're just familiars versus unfamiliars. They're sort of a light gray versus a dark gray. They like the ones that they're familiar cage mates when they're given saline, but they spend more time. Now notice when the oxytocin is blocked, OTA, oxytocin antagonist, when you block oxytocin, you spend less time with your familiars, conceivably either more time in the middle or more time with strangers. Now, the other effect here is that, so this is the bottom panel, this is oxytocin's effect on flocking density. And so this is whether or not you prefer to be with a lot of birds or with not many birds. And so this is also affected by oxytocin. So when you have oxytocin, you want to spend time with increase the number of birds that you're willing to spend time. Okay, so the last one for social behavior, which is what we'll call comforting behavior. So if you put a chick, like a baby bird, like right? That's a baby bird there. Put that in your hands, right? So if you put that in your hands, it does this adorable response where it closes its eyes. It becomes comfortable. Okay? That's what's shown here. And so what we're measuring is latency to close your eyes for 30 seconds. Right? So you have the experiment where they put you in your hands, right? And you're measuring the length of time that it takes for the comforting behavior to take effect. Right? So how long does it take?
50 seconds.
Yeah, less than 50 seconds. Yeah, what, 40 seconds maybe? 35, 40 seconds. I guess chicks are pretty, they're pretty comfortable. Like less than a minute, like stranger holding them, like, okay, yeah, this is great. Okay, so what is naloxone? Does anyone know what naloxone is?
Isn't it used to treat?
That is Narcan. So Narcan is the sort of trade name for naloxone. Naloxone is a mu opioid receptor antagonist, right? It is a competitive antagonist. It is used principally, as is known for Narcan, so if someone has an opiate overdose, right, naloxone is given and it outcompetes the receptor for the opiate itself, right? So it bonds more strongly to the receptor and it displaces morphine, fentanyl, whatever it is, right? Is displaced by naloxone. Okay, so what it means is that it blocks the mu opioid receptor, So what does that tell us about our trusting baby birds?
They are dependent on opioids.
Right, yeah, so this behavior is at least partially mediated by opioid receptors, right? So opioid receptors, right, there's lots of things like endomorphins, endorphins. These are the sort of natural endogenous opiates. And so this behavior, right, their response to comforting behavior is mediated, right, by mu opioid receptors. Right? So because if you block the opioid receptors, then it takes them longer to be comforted. Okay. So this is usually where we stop and talk about, right? We start going over figures that we've talked about in the past. Do we want to do that, or do we want to start like 10 minutes of aggression? I'll let you guys decide.
Aggression. All
What
right.
do you think?
All right.
Let's take a vote. Want to do review for slides, or do we want to do an introduction to aggression? So slides.
Wait, what? Which one's what?
Slides first, like review for figures, or aggression? Right, I guess aggression it is then. All right. These bears are being aggressive. How can you tell?
It's gonna
So,
take his face off.
again, one of our sort of main themes here is going to be agonistic behavior. Aggression occurs in different contexts, and so if we're trying to understand the mechanism, and believe me, I'm not going to go for 30 minutes. We're just going to do an introduction here. And I think it is valuable, right? So notice that you could have, right, agonistic encounters have two components: aggression and submission. And one of the key ideas is that an encounter isn't over until submission occurs, right? So that's a really important idea, right? Because remember, I think from a behavioral perspective, aggression is conflict resolution. And a conflict isn't resolved until both parties agree it's resolved. And the resolution is submissive behavior. So if we thought about mechanisms for this type of thing, you could imagine that you could have essentially one system that oscillated for aggression on one side. And so let's say lots of testosterone on one side means aggression, and low testosterone means submission, right? Something like that. Or you could essentially have two separate systems where you had high and low submissiveness, like you essentially had one system for submissiveness and one system for aggression, right? And so I think while this is not completely resolved for every different type of aggression or agonistic encounter, I think B is generally more likely to be the case most of the time. Now, when we talk about hierarchies, which we'll talk a lot about, that makes a lot of sense. Right? So, and this is especially, so there are different types of hierarchies, but I think most, when we think of a hierarchy, most of what we're thinking about is an alpha, beta, gamma, delta, and so on to down to omega. That's what's called a linear hierarchy. And so one of the things to note about that is that it's really based on submission. Your place in the hierarchy is not based on aggression, it's based on submission. And so what do I mean by that? Let's unpack that for a second. So, who does the alpha submit to? No one. Who does the beta submit to? Who is the beta aggressive to? Everyone else but the alpha. And you can see so on down the line that every time you go down, so if you're gamma, you submit to alpha and beta, but you wouldn't think of submitting to delta or anyone below that, right? So notice that in each of those interactions, you can be a very aggressive gamma, right? But also just as submissive to alpha and beta as anyone could be, right? So from the sort of stereotyped aggression, I think that B is often a better model. But not always, and we'll kind of take a look at that. Now, there's lots of, when we talk about aggressive behavior, there's lots of different types of aggressive behavior. We've already sort of looked at maternal aggression once, but there's a bunch of things on here, right? Like predator aggression, intermale aggression, fear aggression, irritation-based aggression, territorial aggression, right? Predatory attack, instrumental aggression, social conflict. There's lots of different subcategories of aggression. And so, does the cat feel like it's being aggressive to the mouse, would you say?
No.
If you took a cat, like, so, and again, just one way to think about this, we've been talking a lot about dogs, so we'll talk about cats, because this is the model in here, right? In aggression. So, from a neurochemical perspective, do you think the cat feels the same thing being aggressive, attacking and chasing and trying to eat a mouse as it does to fighting another cat? No, right? Those are very different contexts, and as your instinct is correct. We're going to see very different things, and I think from the perspective of the cat, hunting down the mouse, right? The test known as miricide, right? Is probably not going to, like, if we could theoretically ask the cat how it felt, it would not think that it was being aggressive. But from the perspective of the mouse, you could imagine that feels very aggressive, right? We're gonna mostly look at, I'd say the most common paradigm is this idea of the resident intruder paradigm. So essentially the resident intruder is just like it sounds like. You have an animal in its home cage, you add typically a member of the same species and the same sex to their home cage. So the resident is the one whose home cage it is. The intruder is the one who typically will get attacked by the resident at some point. And so that'll be sort of our general model, but as I said, there's a lot of diversity in this group. So, again, I think the easiest way to really think about this is that aggression is conflict resolution. I mean, plain and simple. And anytime there's a limited resource, there's going to be conflict over it, right? This is true from every animal that interacts socially, right? There are no unlimited resources, and so there are conflicts over territories, food, mates, right? And again, an aggressive encounter is not over until submission. Now, in some cases, that submission might be just shy or post-mortem, but the idea here is that the encounter is not over until there is an agreement, more or less, that the encounter is over. All right? Now, this is why I think it's a better sort of framing of this idea to consider agonistic encounters, right? So both the aggressive side and the submissive side within the context of the social interaction. Right? Because again, there is no aggression without a receiver, right? There has to be a receiver, and again, typically when you see an aggressive encounter, there will be escalation until submission is achieved, right? But again, submission really is just as critical as aggression in this context. Now, let's talk about androgens. So much like with male reproductive behavior, androgens are going to play really an outsized role in this. And there's lots of reasons to think that androgens might be associated with aggression. I'm going to tell you right now, know these. Okay? Again, you know at this point, I think you've taken 2 exams, you know it is not my goal to trick you. Right? I want you to understand these fundamentals. If someone says, why do you think androgens are, i.e., why do we think androgens are involved in aggression? Well, when are animals most aggressive? And what do they have when they're trying to mate? Androgens. So there is a seasonal variation in androgen levels that, lo and behold, tracks with a seasonal variation in aggression. During the breeding season, we have high levels of aggression and high levels of androgens. Notice that aggression in males increases massively during puberty, when androgens also increase massively. Now, the other thing to note here is that there is generally, although you said not exclusively, a sex difference in aggression. Which sex tends on average to be more aggressive?
The male.
Males. And which sex on average tends to have more androgens?
The males.
Right? So 3 lines of, I would say, pretty compelling evidence to lead us to suspect that androgens are going to be playing a role here. So, one way. Other thing to kind of keep in mind, if you guys are familiar with deer or not, I don't know if we have any hunters here or not. Maybe we'll spend time outside, the outdoors. You've seen a deer, you know what a deer looks like, right?
Yes.
Yes. Recognize the deer if you saw one running out there? Okay.
Not
All right.
even elk.
So, which sex has antlers?
The males.
The males. And what are those antlers for?
Fighting.
Fighting and impressing females. Okay? Now, a fun fact about antlers, if you're not familiar with them, is that they are shed and regrown every year.
That's great.
Okay? So, there's another point here, which is that every year that they're regrown, they are bigger than they were the year before. So, a male with a big set of antlers is a much older male than the same size male with a smaller set of antlers. Right? The size of the antler is indicative of the age of the male. Now, we say, what are antlers for? This is what antlers are for. Now, these are red deer. Are red deer monogamous?
No.
No. What's a good general rule of thumb to identify whether we're looking at an animal that's monogamous or not? That's not universally true, but what's a good indicator that you're looking at a monogamous animal? Sexual dimorphism, they look the same. If you can't tell the difference between a social animal, right? Now again, I say that because you can't tell the difference between snakes, but they're not social, right? Asocial animals, the males and females look the same, or a lot of times they look the same. But if they are a social animal that looks the same between males and females, odds are they are monogamous and probably biparental. No, these guys are not. Now, what they do is that they carve out territories. Males fight other males, and the best males get the best territories. The territory is a physical space, a fairly large physical space, in fact, right? So they carve up these spaces during the breeding season, and then females, what are females choosing here? How do they know who is the best male?
The antlers.
That's one good indication. The biggest and best territories are owned by the biggest males, and typically the largest set of antlers is on the biggest male. He has the best territory. And why do the females care about territory? Where they eat and where they raise their young. So the bigger the male's territory, the more females and the more, for some reason I'm blanking on the name of a baby deer.
Fawn?
Fawn? Right, they're fawns, right? Am I
Think so.
mistaken? Yeah, so the more fawns that can be supported on that territory. And so the basic idea here is that the bigger your territory as a male, the more females and offspring that you can support. And so from the female's perspective, notice that it's better to be like the 5th female on the best territory than it is to be the number 1 female on a crappy territory. Right? And so this is what gives rise to polygamy. The females and the males are operating in their own reproductive best interest. Now, as you can imagine, red deer are quite feisty during the breeding season. And the mating season in these animals is actually in November. And so what we're looking at here is the change in testosterone, antler growth, and aggression. Notice that testosterone precedes all of these things, and testosterone, antler growth, and aggression pretty much peak at the same time right before the mating season. After the mating season is done, notice that we have a drop in aggression, a drop in testosterone, and eventually we will actually physically drop off our antlers altogether. Right? And then we will do that until we go around the next year when we do this all again. Now, what we have here is clever scientists doing these manipulations. So notice that, oops, we're going the other way. So this is the winter, right? December, January, February, March, right? This is winter. We're just coming out of it. It's supposed to be spring, but yesterday it certainly didn't feel like it. So we're going to do a few things. We're going to castrate them in the winter. We're going to give them testosterone implants in the winter. We're going to give them testosterone in the summer. And then we are going to amputate their antlers altogether. So if we castrate them in the winter, what will we predict happens to their social rank? It's going to plummet, right? If we give them testosterone in the winter, they are going to retain their antlers for longer, and unsurprisingly, they're going to be much more aggressive. If we give them testosterone in the summer, right, if we look back at this, right, notice that the summer, here the testosterone's going up here, we are going to make what essentially amounts to a social climber, right? It's going to be massively increasing its status, and obviously if you amputate the antlers off of a deer in the breeding season, they will suffer an immediate loss of social status, okay? So testosterone is related to social status in the sense that it works at the level of the brain to promote aggressiveness. So that aggressiveness tends to lead to being higher social status, right? It actually increases antler growth itself, right? So if we add testosterone, right, we are increasing the ornamentation of the male. And this is primarily an effect of that occurs mostly in the breeding season, where we get changes in tissue responsiveness to androgens, and we also have females being around, right? And so you get this relationship between testosterone and aggression specifically manifests itself in the breeding season. So I'm going to stop there, right? And yeah, we're done. It was a long one, I know, but these figures are complicated, right? So I know, especially that one with the antagonists, right? With the different brain regions and all of that. So make sure that you go over it when I'm not there to explain it to you, and make sure that it makes sense, right? And so try again to sort of write those out, and if you have any questions, we'll kind of resume our last 30 minutes of figure review on Thursday, when we're talking, it'll be all about aggression, and one of my favorite animals that got taken out of this book, the hair sparrows, which are super fun. Ooh.